I never made it, but I know what it takes. I'm motivated by a mix of emotion. Got my statement, and I'm reading it slow so I can understand it fully. Appreciate my standing, go out of this world and right on time. Oh, never in my life have I seen the sky light up like this. I'm far away from Earth and I wanna know if you exist. Mission control, do you read me? Send me my coordinates lately. Mission control, am I drifting? Tie me to someone who will love me. Yeah, wait, I think I'll last a bit longer. Find another reason to breathe this air. And now we made for ourselves. This won't be the last time. Oh, never in my life have I seen the sky light up like this. I'm far away from Earth and I wanna know if you exist. Mission control, do you read me? Send me my coordinates lately. Mission control, am I drifting? Tie me to someone who will love me. Yeah, I knew it when I met you. I'm not the only one who's alone. Find me soon and take me home. I knew it when I saw you. I'm not the only one hanging on. Tie me to someone who will love me. Oh, never in my life did I wanna see you more than this. And every time you're lost, I wonder if you know you're missed. I'm breathing when I can, and I only hope you're watching this. I hope you know that I exist. Mission control, do you read me? Send me my coordinates lately. Mission control, am I drifting? Tie me to
Indonesia is an archipelagic country with one of the world's richest reefs and longest coastline. This gives Indonesia an immense potential in the fisheries and marine sector. The Faculty of Fisheries and Marine Science of Diponegoro University has been among the leading marine research institutions in Indonesia since it was first established as Fisheries Department in 1968 and officially became a faculty in 1994. The Faculty of Fisheries and Marine Science is located in the Central Java province. The main campus, where main activities take place, is located in the capital city of Semarang. The second campus, also known as the Marine Science Technopark and Coastal Development Laboratory, is located in Jepara Regency. The vision of the Faculty of Fisheries and Marine Science is to become an excellent faculty in the field of fishery science and tropical marine in the year 2020. In pursuing its vision, the Faculty of Fisheries and Marine Science missions are to provide a high-quality education in the field of fisheries and marine science, engage in scientific research and publications, produce intellectual property rights, provide community services in fisheries and marine science field, implement a professional and accountable system, and empower students' activities to achieve the complete graduate qualifications. There are 6 departments in the Faculty of Fisheries and Marine Science. The first one is Aquatic Resource Management, Capture Fisheries, Aquaculture, Marine Science, Oceanography, Fisheries Product Technology. The faculty provides numerous facilities to support all study and research activities, from student discussion spaces, library, and integrated laboratories. We provide aquaculture lab, biotechnology lab, chemistry lab, hatchery, and geographic information system lab.
The excellence of Faculty of Fisheries and Marine Science is proud to contribute in fisheries and marine field through artificial patch reef in Panjang Island, Jepara, provides information to the public of Semarang as a reference data for sea level rise on the coast with drop calendar. In our faculty, we collaborate academic and experimental learning to give the best experience of learning. The faculty have 16 student activity units divided into several fields like art, sport, religious club, research, student executive board, and student senate, where students are allowed to become more involved on campus, increase leadership, social responsibility, and their personal development. One of the organization, Kesemat, received an achievement as the best ASEAN student organizations in 2016. Faculty of Fisheries and Marine Science conducts activities in international scale, such as International Tropical Summer Course and International Conference on Tropical and Coastal Region Eco Development. International Tropical Summer Course is an event for international students from partner universities to learn about coastal ecological preservation and coastal biota in Semarang and Karimun Jawa Island. International Conference on Tropical and Coastal Region Eco Development is an annual international seminar event that aims to promote and disseminate research results.
The Faculty of Fisheries and Marine Science aspire to take a role in strengthening the marine and fisheries field in Indonesia through the application of knowledge from our graduates to achieve and bring positive impact to the society.
Indonesia merupakan negara kepulauan terbesar ke-6 di dunia, dengan garis pantai, pulau-pulau kecil, dan pesisir yang sangat luas. Beragam kekayaan sumber daya hayati perikanan sangat potensial untuk dimanfaatkan. Ada plankton, ikan, krustase, moluska, tumbuhan air, karang, echinodermata, dan lain-lain. Tugas kita semua untuk dapat mengelolanya dengan bijak agar lestari. Di program studi manajemen sumber daya perairan FPIK Undip, kalian akan mendapatkan bekal untuk menjadi sarjana perikanan dan SDM yang mumpuni dalam melestarikan sumber daya perairan dan perikanan. Kajian yang dipelajari dari mulai lautan hingga pesisir, seperti ekosistem terumbu karang, padang lamun, mangrove, estuari, sampai ekosistem air tawar, seperti sungai, danau, dan waduk. Program studi ini 4 kali berturut-turut memperoleh akreditasi A atau sangat baik. Sejak berdiri pada tahun 1996, alumni MSP telah tersebar di seluruh Indonesia dan bekerja di berbagai instansi seperti Kementerian Kelautan dan Perikanan, Kementerian Lingkungan Hidup dan Kehutanan, LIPI, pemerintah daerah atau dinas, institusi pendidikan, perbankan, lembaga pendidikan, dan menjadi entrepreneur. Dukungan tenaga pengajar yang kompeten, sarana prasarana laboratorium yang lengkap, menambah keunggulan prodi ini. Di prodi MSP, kalian dapat melanjutkan jenjang studi master dan doktor yang inline, serta memungkinkan untuk berinteraksi dan berkolaborasi dengan jejaring universitas luar negeri. Pada kurikulum Merdeka Belajar saat ini, kamu bisa mengikuti pertukaran mahasiswa serta magang di berbagai instansi yang bonafit dan keren pastinya. Jadi, kamu akan mendapatkan banyak pengalaman yang bermanfaat dan pastinya akan membuat kamu menjadi pribadi yang tangguh. So, tunggu apa lagi? Yuk bergabung dengan kami di Prodi MSP FPIK Undip. Informasi lebih lanjut, hubungi kami.
think I'll last a bit longer. Find another reason to breathe this air. And now we made for ourselves. This won't be the last time. Oh, never in my life have I seen the sky light up like this. I'm far away from Earth and I wanna know if you exist. Mission control, do you read me? Send me my coordinates lately. Mission control, am I drifting? Tie me to someone who will love me, yeah. I knew it when I met you, I'm not the only one who's alone. Find me soon and take me home. I knew it when I saw you, I'm not the only one anymore. Tie me to someone who will love me. Mission control, do you read me? Send me my coordinates lately. I'm never in my life, am I drifting? Tie me to someone who will love me, yeah. I knew it when I met you, I'm not the only one who's below you. Tie me soon and take me home. I knew it when I saw you, I'm not the only one anymore. Tie me to someone who will love me. Mission control, do you control? Do you read me? Send me my coordinates lately. Mission control, tie me to someone who will love me. I know it when I met you, I'm not the
Universitas Diponegoro atau Undip merupakan salah satu universitas terbesar di Indonesia yang terletak di Jawa Tengah. Undip memiliki 11 fakultas dan telah memiliki 141 program studi. Berdasarkan data Kemenristek-Dikti, Universitas Diponegoro menempati peringkat 6 dari 13 perguruan tinggi terbaik di Indonesia tahun 2019. Menurut World Green Metric Ranking, Universitas Diponegoro menempati peringkat 78 dengan skor 7025 yang merupakan peringkat 3 se-Indonesia. Fakultas Perikanan dan Ilmu Kelautan Universitas Diponegoro berdiri pada tanggal 8 Oktober 1968 yang merupakan pemekaran dari Fakultas Peternakan dan Perikanan. Saat ini, Fakultas Perikanan dan Ilmu Kelautan memiliki 6 departemen, terdiri dari 6 program studi S1 terakreditasi A, 2 program studi S2, dan 2 program studi S3. Program studi Ilmu Kelautan, program studi Oceanografi, program studi Aquakultur, program studi Perikanan Tangkap, program studi Teknologi Hasil Perikanan, program studi Manajemen Sumber Daya Perairan, S1 Manajemen Sumber Daya Perairan, S2 Manajemen Sumber Daya Pantai, S3 Manajemen Sumber Daya Pantai. program studi manajemen sumber daya perairan. Tujuan dari manajemen sumber daya perairan yaitu diharapkan mampu menguasai dan menerapkan prinsip-prinsip dan mampu mendayagunakan sarana dan prasarana guna melaksanakan pengelolaan sumber daya perikanan dan perairan. Program studi manajemen sumber daya perairan memiliki laboratorium pengelolaan sumber daya ikan dan lingkungan yang terdiri dari sub-laboratorium hidrologi, sub-laboratorium ilmu-ilmu perairan, sub-laboratorium pengelolaan sumber daya perikanan. Kegiatan praktikum program studi manajemen sumber daya perairan. Prestasi mahasiswa program studi manajemen sumber daya perairan. Selain dalam bidang akademik, program studi manajemen sumber daya perairan melakukan kegiatan pengabdian kepada masyarakat.
Yugo and from Japan, and I'm really enjoying this ITRO School 2019. I enjoyed the whole stay in Semarang and Karimunjawa Island. That was a real beautiful island. I really enjoyed this program. Thank you.
For ITROSCO 2019, we are so happy to be here, to be part of ITROSCO family, and then we would like to say thank you to the organizer, and then to all the committee. They are so superb, the food are so delicious, and then
thank you for the next level hospitality. As well, the divers who managed to train and accompany us during the snorkeling activity in Karimunjawa.
I appreciate all the programs that you had done for us, and I hope that the program will benefit us in future.
And I can say, even though we just knew each other about 10 days, it feels like 10 months.
I think it's a great course because I can compare with maybe Sweden, maybe Indonesia, maybe Japan, maybe Malaysia, their culture and their language. We can find a lot of funny things between different culture. Yes, I suggest all the people will come here to join this club.
The ITROSCO trip has been really cool, really interesting. My favorite part was the trip to Karimunjawa because we got to go snorkeling at coral reefs, and that was just an incredible experience seeing it up close. In Indonesia, it's been really interesting. People have been so hospitable, and then in this program, it's been very cool. It's just fun to hang out with people from different backgrounds. When you're all doing it together, it makes more connections, which is sweet.
It was nice bonding with other students from Malaysia, Taiwan, and Japan. We also had some lectures towards the beginning, which taught us about mangrove conservation and ocean conservation, and it really stressed the importance of trying to save our biodiversity. We also learned a lot about land subsidence and that problem in Semarang. And I think I really had a great time. The highlight of the trip was definitely the excursion to Karimunjawa.
It was really fun to be able to be in the ocean in Indonesia with the crystal clear blue water and see the coral. Karimunjawa was really fun, and it's been great being able to meet people from all different cultures. Coming from the
Assalamualaikum warahmatullahi wabarakatuh. Hello, good morning.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, everyone. Especially good morning from Semarang, Indonesia. Welcome to all of you on today's webinar. With the topic is Unveiling Stream Diversity in the Aquatic Environments, their Taxonomy and Conservation. The Honorable Dean, Faculty of Fisheries and Marine Science, Universitas Diponegoro, Professor Insinyur Triwinarni Agustin, MSc PhD, the Honorable Head of Department of Aquatic Resources, Dr. Insinyur Suryanti, MPI, the Honorable Secretary of Study Program Aquatic Resources Management, Bukurun INSPI, MSI, the Honorable Head of Postgraduate Program of Aquatic Resource Management, Dr. Anindya Sabdaningsih, SSC, MSI, the Honorable Speaker on this webinar, Dr. Push, from Center for Marine Living Resource and Ecology, Government of India, and also to all of participants on this webinar. Allow me to introduce myself. My name is Kuku Prakoso, as Master of Ceremony on this webinar. Before we start, allow me to read an agenda and rules for today's webinar. Maybe the committee can help us to show the screen. Yeah, this is the agenda of this for today's webinar. There are two sessions, especially from Dr. Pus. At the first session, he will talk about the taxonomy of shrimp, and the second session, he will talk about the conservation, and the last we have a discussion, of course, and closing. And the next is the rules for this webinar. The seminar is held through two channels. There are Zoom and YouTube. For participants who have difficulty logging into Zoom, can switch to YouTube. And for the stability of the internet connection, participants' video and audio are automatically turned off and will be pleased to turn on into the photo or discussion session. In this discussion session, please use the chat feature on Zoom to ask questions. Those who want to be conferred directly can use the raise hand feature. For participants who are present on YouTube, can text question or live chat. Then the attendance link will be shared through the chat menu on Zoom and live chat on YouTube during the event. And participants must attach proof of attendance, such a screenshot of Zoom and YouTube subscribes. Okay, thank you for the committee. Move on to the next agenda. We will have an opening remarks by our Dean, Faculty Fisheries and Marine Science, Universitas Diponegoro, Professor Insinyur Triwinarni Agustini MSc PhD. For Professor Triwinarni Agustini, time is yours.
Okay, thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Yeah. Good morning, everyone. And first of all, I would like to thanks to God for having us together to be here in a online program with the webinar, and this is very good topic that will be discussed for today. And here I would like also to thanks to the honorable guest lecturer, Mr. Dr. Paramasipam Purusottaman from Center for Marine Living Resources and Ecology from Cochin, India. Thank you very much, Mr. Pus.
Thank you.
This is the nickname for him. Thank you for keeping time and sharing your experience with us. And our honorable head of department and secretary of department, Dr. Suryanti, and also Kurun Ain, MSE. And also we have a moderator here, will be Professor Dr. Norma Afiati, MSc. Thank you very much, Prof. Norma, for having your time. And of course, for today, because we will discuss about the unfailing shrimp diversity in the aquatic environment with focus on the taxonomy and conservation. It is very important topic that will be discussed together, and maybe Dr. Pus will sharing their experience for all of us. And this is very good because shrimp is the most important commodity at the moment from Indonesia, and we have a huge export to other countries. And the diversity for the shrimp is very great, and it is why we need to discuss which kind of shrimp that need to conserve for the next future, because it is really need to do conservation for the species that we need to aware, all of us. So I think we also hope that Dr. Pus or Paramasivam sometimes have chance to come to our university or our faculty, especially to share and meet face to face with us. This is our hope, and because one of our targets to achieve that more invited speaker for abroad that has a qualification, especially minimal for PhD degree, so it can be counted as the output for our achievement, especially for the faculty to become reported by university as the world-class university. So this is a good chance, and hopefully other collaboration will be followed after this event. So I think that we really welcome and thanks for all of the arrangement on the committee for having this program. So thank you very much, and have a nice discussion and fruitful result from this event. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Prof. Thank you so much to Prof. Triwinarni Agustini for a great opening remarks for our webinar today. And we move to the next agenda. We will have a photo session. I kindly asking for every participants in this webinar can open their camera so we can capture this moment for a second. And for the documentation, the committee will help me to take a picture. So maybe all of you can open your camera and smile gratefully. For the committee, please help me to take a picture.
Okay, Mr. Kubo, I will take a picture for the audience. Please, the activity on camera. I will take a picture. 1, 2, 3. Okay, next slide. 1, 2, 3. 1, 2, 3. 1, 2, 3. 3, and the last slide. 1, 2, 3. Thank you, Mr. Kobo.
Okay, thank you so much, Mr. Fian, for your help. Okay, we will move to our next agenda. Finally, we move into the key point of today's session. There is start from Dr. Bruce, but before that, to kick off today's webinar, allow me to invite the moderator, Professor Norma Arfiati, MSc PhD. She is one of professor in our Department of Aquatic Resource, which lead this webinar. Hello, Prof. Norma.
Hello, Mas Kuku, how are you?
I'm fine. I'm fine, Prof. How are you today, Prof?
Okay, me too. Thank you. Thank you for everything.
Yeah, thank you so much, Prof, to be able as moderator for today. And I kindly asking you to lead this webinar, Prof. Yeah.
Okay.
Okay.
Yeah,
Time
I
and
would
screen
like.
are yours, Prof.
Okay. Yeah, I will share screen for. Okay. Can you see this, Mas Kuku? Can you see my slide?
Yes, Prof. It's clear.
Okay. Yes. Well, once again, good morning, everybody. Good morning. Vidin is still with us here, and also some of the management. And good morning, all the participants, and especially our honorable guest for this morning, that is Dr. Paramasivam Purushottaman. For short, he said to us that we may call him just simply Push. We have been talking to him yesterday, actually last night, and in both countries, in Indonesia and in India, Puss is cat, more or less like that. Okay, so I would like to introduce Dr. Paramasivam Purushottaman, the one who is going to lead this general lecture, Unveiling Shrimp Diversity in the Aquatic Environment, their Taxonomy and Conservation. No, this already. Let me. Hang on. Yeah. Oops. Yeah. I would like to draw your attention on the expertise shared today by Dr. Push. He has an expertise in several fields of the area, especially in shrimp and lobster. The first is the shrimp and lobster taxonomy, and he has an expertise in using line drawing GIMP tools. Yeah, expertise in molecular taxonomy on marine species, also the characterization and data analysis of microsatellite marker, also expertise in marine biological modeling using the multivariate state statistical method. Also, he is very good in phylogenetic analysis using various bioinformatic tools, which is very important if you are studying biomolecular in various kind of organism. He has also a solid scientific proficiency. He is able to collaborate well with various people, and he has very independent thought processes. You will see later that he writes lots of research proposal and lots of publication in peer scientific journal. This one. Considering the education qualification of Dr. Purushottaman, in 2019, he awarded a Doctor of Philosophy in Biosciences from the Mangalore University in India. His thesis title is Integrative Taxonomy of Deep Sea Shrimp, Aristos Alkoki Ramadan, resources along the southern coast of India. This title is awarded in 27th of February 2020. He has Master of Science in Biotechnology from the Department of Biotechnology, Baradidasan University in India. For that he spent 2 years to get the title, and his Bachelor of Science is in Biotechnology from Tiruvallur University, India. For 3 years, he's been studying from 2007 until 2010. Wow, he has lots of professional and research experience, at least 1, 2, 3, 4, 5, okay? Well, at this moment, in the year 2020 to present, he works for the Center for Marine Living Resources and Ecology in Cochin, India. Before that, he works for several years in National Bureau of Fish Genetic Resources, also in Cochin, India. Later, before that, or prior to that, since 2013 until 2018, he works for Central Marine Fisheries Research Institute in Cochin, India. And starting half a year, I think this one, in 2012, he works for the National Institute of Oceanography, Cochin, India, for which he was pursuing his master dissertation. And by doing so, he also trained in the immunology laboratory in that National Institute of Oceanography. At least, as the first author during these last 5 years, Dr. Paramasivam already has 1, 2, 3, 4, 5, 6, 7, 8, 9. Actually, it's 10. I think I missed one of it. So you can see that he has lots of publication in a very high reputative journal, in Nauplius, in Zootaxa, in Journal of Genetics, and also in Zootaxa, in Journal of Zoology, Journal of Crustacean Biology, and also in Zootaxa. This is very astonishing, at least for me. And I think for many of us here in Indonesia, he is a very productive young researcher. And beside of those 10, he has at least 30 other publications in various high qualified journals within these 5 years as a collaborator to other scientists. And besides of that, he has 3 articles under communication or under review in 3 different journals, in reviews in aquaculture, Zootaxa, and marine biology research. And I think all of them is about decapod, or is about crustacean. That is my introduction for Dr. Paramasivam Purusotaman. I would like to stop share, please. Thank you. And now I think it's time for us to listen to our honorable guest lecturer, Dr. Paramasivam Purusotaman. Time is yours. Please.
A very good morning, Professor Nurma. So also very good morning to Professor Trai. So first of all, I will thanks to all the participants and Professor Trai, and especially for Professor Norma, so giving the opportunity to present my experience on shrimps taxonomy. So here today we will discuss about an overall diversity of the shrimps and their applications, and then also that some of the slide about the conservation, how we can conserve, then how we can manage that resources in the sustainable way. So today we will discuss it in detail. So first I will share my slides. Is it visible?
Yeah.
Yeah, we'll start. Once again, very good morning to all. today we'll see that in the topic of unveiling shrimps diversity in the aquatic environment and the taxonomy and conservations. So nowadays that especially in the classical taxonomy, people are reduced because of there is time taking subjects, and also it's very much patience is required for the classical taxonomy. So here, that's why I'm here to give some interest to the coming generation. So in the presentation, I thought it will give some interest on the classical taxonomy of shrimps group, because shrimp is one of the important group in the crustacean group, so which is mostly commercially used in the capture fishery, at the same time an ornamental purpose also. So here today the presentation is covering in 5 different topic. First, we will see in detail about the species diversity of shrimp groups. Next, we will discuss about the integrative taxonomy, both classical and conventional, classical and molecular taxonomy we will see. And in between, we will discuss ecological role of the shrimps, especially in the aquatic environment. So the shrimps, what role is doing in the environment, we will discuss it. And further, we will see in detail about fishery and ornamental application of the shrimp resources. Finally, we will discuss about the conservation of the important natural resources. So this is the topic I'm going to cover today. So first, we will see that species diversity. How many species are there in the overall aquatic environment? Today, first we will see it. Actually, the shrimps is one of the important group in the crustacean group, which is having a very large number of species in the group. If you see in the size ranges, which is it is starting from microscopic to macroscopic, around 35 centimeter long. So at the same time, the species are mostly commercially important species, both commercial fishery, at the same time ornamental purpose also. So the shrimps mostly distributed all the environment, freshwater, brackish water, marine environment. But however, that major diversity it is present in marine environment. So at the same time, which are living in different depth bottoms around under the rock, mud, and the sand. Then under the coral environment also, these species are commonly distributed. Actually in the fishery, there is a 2 terms are commonly used for the this group. One is shrimps and prawn. Especially in India, the most of the marine species we used to call the shrimps, and freshwater species we used to call the prawn. However, both the terms are mostly common, and there is no specific description for this species. But in internationally, all the species we simply used to call the shrimp only. So that is the accepted name in the trade and also the worldwide. So coming to the how many species in the shrimp group, so almost more than 4,050 species are still now described. However, it will be remaining around some 10,000 species will be available. So, some of the researchers are suggesting. So currently, more than 4,050 species are described, which are mostly four different groups. The major group is Caridia, which is having more number of species, around 3,500 species are available. The next important group is Dendrobranchiata, which is having around 550 species in this group. So other 2 groups are small groups, Cynopodia and Procaridia. So these are all the groups is available in the shrimps group. So then about the morphological variation, we will see in coming slide. So here, this is the diversity of shrimp group. So next we will see about the taxonomy of shrimp group. So the before starting that taxonomy, first we used to know that morphology of the shrimps, especially the shrimps having a different structure to other organisms. So as we know that the taxonomy, the father of taxonomy called Linnaeus started that journey for identification of the species through the morphological way. And especially in crustacean group, the Spence bait is involved and described more number of species in the shrimp group. So in unofficially we can consider that Spence bait is the father of shrimps taxonomy, because who has described a lot of species, and he has collected many of the species from HMS Challenger expeditions. So we can consider Spence bait is the father of shrimps taxonomy. So coming to the morphology of shrimps, shrimps laterally compressed elongated body, which is having a 19 segment in the 19 segment in the body. So here the major part are one is carapace, other one is abdominal segment. The carapace known as it's a mixture of head and thorax region. So altogether we called as carapace. So here the shrimp morphology, there is a different anatomical terms are used. So here we can see the different name on different part. We can see that. So one of that carapace. The carapace is having mixture of head and thorax. We're having around 13 segment, which is intact inside of the carapace. In other important part is abdominal segment, so which is having a 6 segment. So altogether, totally 19 segment. It is starting from antennule to tail regions. So the 19 segments are antennules, antenna, then mandible, then maxilla 1, maxilla 2, then further maxillipede 1, 2, 3. Then it will start that periapod, which is a walking leg. Simply we called as walking leg. So this is the walking legs. It will be there under carapace regions, and then now further it will be pleopod, that is used for the swimming purpose. It is we can call as swimming legs. So here the name is pleopods. Here the 5 pleopods will be there. And finally uropod. The tail portion we called as uropod with the telson regions. So these are all the morphological part is very important for the species identifications. Also that the major morphological features are occur mostly in the carapace itself. So here we can see so already I told you that carapace is a mixture of head and thorax region. Here having the many morphological part is based on that where it is present that specific morphological features which is hanging the regional specific name, especially in if any specific character, example, any groove is there in the cervical region, so they called as cervical groove. So suppose a spine it is present in a hepatic region, they called as hepatic spines. According to the sulcus or a groove present in the particular area, the most of the names are called with the based on the region itself. So this is the very important part for the morphological identification, especially rostrum. The rostrum in dorsal region, there is a teeth will be there. The based on the teeth count and the teeth pattern, it will be helpful for the identification. Then other important features is periopods. The periopods, that means the walking leg. So having a two different structures. So one is the exopod, other one is the endopod. The exopod will be there inside, and endopod will be extended with the 5 segments. Here you can see that the endopod, this is the endopod, so which is having a 5 segments, ichium, meatus, carpus, propodus, and finally dactylus. So these are all the the segments are present the periopod, which is very important, which is having a specific character, especially the pattern of spine and other morphological features will be present in the periopods. So here, especially uh, inside of the carapace, each periopod having some other extra characters. Those characters, especially the gill region, having 3 variety of gills, so that it will start from podobranch and arthrobranch and fluorobranch. When that initially starting from podobranch, when the gillet is present near the coxa region. This is the coxa, that is the below of base. So this is exopod and endopod here in below that base. So which is starting both exopod, endopod from base. Then below that we used to call as coxal plate, which is transparent transparent sheet-like structure, which is having that gill structure too. So here, based on the arrangement, they say they called us podobranch and arthrobranch and fluorobranch. These are also very important for the segregation of this shrimp group. In further, the antennules, this also is segmented with three article, and also having a 2 different variety of flagella. And finally, the tail portion, especially tailson, having a specific character for many of the species. So the both fixed finger and movable, fixed spine and movable spine will be present. The few of the pinnate groups, so most of the pinnate groups having both fixed and movable spine will be present. So these are all the general terminology used in the classical taxonomy of shrimp group. Yeah, here we will see that variation on the 4 groups. Already I told you that in shrimps having the 4 group, the most of the species are classified in that 4 group only. The first one is Dendrobranchiata. So the major order is Pinnidia. In Pinnidia, the Pinnidia shrimps having a specific character, which that is the 3 major character is very important for the classification of Pinnidia. So here we can see that the first character is the, I told you that a periopod will be there here, first 5 pair. So you can see that pair 1, pair 2, pair 3, which is having chelated leg, which means that the propodus and dactylus look like a finger and thumb structure will be there. So altogether we used to call it as chela. So this leg mostly helping for that walking, and at the same time taking that prey item to the mouth. So these 3 legs very important for that purpose too. At the same time, which is having a specific character for Pinnidia. The first 3 legs having the chelated one. So the next important character is abdominal segment. So the abdominal segment, the first abdominal segment is overlapping to the next, and the second is overlapping to third. Subsequently, it's overlapping. So the next character is sexual organ. The Pinnidia shrimps having a specific sexual organ. For male animal having a patasma, that is for a male animal organ, and female animal having a sexual organ is we used to call as skeleton. So later I will show that sexual organ structures. So that based on the structure we can identify the species variation between the Pinnidia also. So next very important group is Caridia. So here that how it is differing from Pinnidia we can understand is the first one is the same periopods. Instead of chelated leg, 3 leg, here only first 2 pair only chelated. So others are simply spiniform structures. So that is the one of the major characters. And the second character is we can identify very easily whether the species is Caridia or shrimp. That is the second abdominal segment, which is overlapping to first and third segment. So this is the thing. We can see that this is the second abdominal segment is overlapping to first at the same time third. So when you see in the pinnae groups overlapping only second, so and third, there is no overlapping to first. So this is the major difference between this Pinnidae and Caridia group. At the same time, this group is not having a specific copulatory organ, which is having a secondary sexual character only. So there is no specific sexual organ. At the same time, after the copulation, the female animal used to carry the egg under the abdominal segment up to the hatch out. But in Pinnidae groups, so after the copulation, used to release the egg directly into the water. So this is the major difference between these two groups. So based on these three character, we can segregate into the whether it is a Pinnidae groups or a Caridia group, we can find out. So next other smaller group is Stenopodia and Procaridia. Stenopoid almost is a mixture of Pinnidae and Caridia. At the same time, we can segregate it from those two groups. Here, the first three periopod will be chelated. At the same time, the segment region overlapping frequently to next, and along with that, the species not having the specific copulatory organ. At the same time, this also used to carry the egg under the abdomen. Okay. At the same time, one more specific character is there for Stenopoid, which is having a minute spine over the body. So the one character is enough to find out whether it is Stenopoid or other groups. The spine itself, we can understand the species are Stenopoid groups. So other very smaller group, which is having only 6 species in internationally, that is Procaridia, which is having a specific character is all the periopod not chelated. All the periopod not chelated. At the same time, the second abdominal pleura, which was greatly expanded to first and third. One more important character is maxilliped. I told you that these first 5 periopods for the walking leg, the next this portion. So this is we called as a maxilliped, which will be used for that taking food from the environment to the mouth region. So that is acting like a hand. So this region having a 7 segment. But compared to other groups, maximum those groups are having a 5 segment, especially in Procaridia having a 7 segment. So based on this character, easily we can segregate our species belongs to Procaridia groups. So this is the major difference between these 4 groups. In further, we will see that morphological variation of Pinnidae groups. Because the Pinnidae groups are very important in the capture fishery, because the many of the species, around 550 species are available in this Pinnidae group. So most of the species are commercially important, which will be used for the edible purpose. So therefore, it's getting highlighted in the fishery, and so there is a required to identify the species in both morphological and classical and molecular way. So very simple character is enough to classify these Pinnidae groups. So totally 5 families are there in the Pinnidae groups. Those very important groups, Solnoseridae, Pinnidae, and Narisidae. Others 2 groups are having very less number of species. Especially Pinnidae is a very important group, both freshwaters and marine waters. Many of the species are commonly used for that food consumptions. So especially how we can classify that these groups in the through the morphological way. The simple few of the characters enough to classify it. For Aristidae, the species which is not having the post-orbital spine, that means, so the orbital regions behind that there is a spine will be there. When the spine is present, we can conclude that the group is Pinaide groups. So, at the same time, when that post-orbital spine is absent, we can go for the other groups. Aristidae is not having that post-orbital spine. At the same time, all the species from the Aristidae groups are having 3 rostral teeth on the dorsal region of rostrum. So this is a very important character for the Aristidae. At the same time, for the Solnoseridae, a very specific character is there, that is the prosartima. That means it's a eye brush. Actually, for other groups, it's not having this kind of character. So especially for Solnosera having a specific character, that is one extended brush-like form in upper region of the eye, so which will be used for the cleaning purpose of the eye. So that here the in taxonomic name is prosartima, which is important for the Solnoseridae family. When the prosartima is present, we can simply use to call it as that is the Solnoseridae group. So if it is Aristidae, all the species from Aristidae are distributed only in deeper water, more than greater than 200 meter only it is present. But Solnoseridae is present both shallow and deeper water. Of course, in Pinaide also it will be then freshwater to marine water. So this is the 3 major Pinaide family in this group. Next, we will see some of the important commercially important species, how we can identify through the morphological way. So first we will see about Aristeas salcage. So it's a very interesting species from the Indian coast, also which is distributed along the Gulf of Aden to the Andaman Sea. So commonly distributed in generally we used to call this Arabian red shrimp, especially in the Indian fishery. The local people used to call as red ring because this species in the fresh condition, so very reddish in color in abdominal segment, that is their ring. So this region is having a very reddish in color with the ring formation. So here the local people used to call as a red ring. So coming to the taxonomy of the species, we can the major characters already I told you that rostrum having 3 rostral teeth with a very reddish in color. When you see like this character, we will segregate that is Aristidae group. Specifically for Aristeas salcage, we can identify based on one microscopic character, that is we have to see under the microscope, in especially the structure of the fluorobranch. I told you that gill region having 3 different structures. One is podobranch, arthrobranch, and finally fluorobranch. So here Aristeas salcage having a particular shape, that is the fluorobranch is a very minute microscopic which look like a spine. Another one closely related species is there, Aristeas semidentatus, which is very close to the Aristeas salcage, almost look like similar, but the major variation is the fluorobranchia is a little lengthy with a leaf-like form. So this is the major difference between these 2 species. Little difficult to see in the naked eye, but we can see under the microscope. So this is one of the very important resources, and which is catched in annually around 2,000 to 4,000 ton in the Indian coast. So also one more specific character is there in Aristidae. All the species having a sexual dimorphism. That means the morphological variation is there between the sex. So when you see the male animal very smaller than female, at the same time the rostrum is very short. If you see in a female animal, that rostrum will be very long and slender, but in male animal very short. So this is the major difference between that sexes. So on the Aristotic group. So next we will see and another one important species from the Pinidae family. So the genus is Metopinopsis. Generally used to called as velvet sheen because the carapace regions having a velvet structure. So because of that character, and commonly we used to call this velvet sheens. So this also one of the important species in the captured fishery, which is having a specific character in the sexual organ. So already I told you that in Pinidae group, most of the species are having the species specific character in the sexual organ. So that when describing about the group level variation, I informed that there is a separate sexual organ is there for Pinidae groups. So this is the male sexual organ. We used to call it as petasma. This is a thylacum, it's a female organ, so female sexual organ. So this is the difference between male and female species. At the same time, every species having a specific character in the sexual organ. So coming to here, the Metopinopsis andamans is having a specific character is asymmetrical petasma. That means the petasma, both the side will be asymmetrical. That means unequally it will be available. So at the same time, the structure of the petasma, there is a extra overlapping lobules will be there. So based on this structure, we can identify that is the Andamans, Metopinopsis andamansis. One more species is there very closely related to andamansis. So that is called as coniger. Almost all the morphological characters, coloration, everything same except for the sexual organ. When you see in the Andamans is especially in petasma is more longer than the half of the carapace. So they compared the carapace length with the petasma. The petasma length is more than half of the carapace, but another one which is coniger is less than half of the carapace. That is one of the important character. But in female, easily we can identify it based on that presence and absence of flap. When you see in the Andamans is having a extra flap on the thylacum, but in the coniger there is no overlapping. So that is the major variation between these closely related species. Both are commonly distributed in the Arabian to Bay of Bengal water with more than 200 to 500 meter depth. So another one species from the Metopinopsis, the same group, which is distributed in the intertidal regions of coral regions, which is also called as velvet sheens. At the same time, which is having a specific character, a morphology, coloration is very specific character for this species, which is covered with dark brownish to white dot over the body. So that is one important characters. At the same time, the specific morphological features are present in sexual organ here too. So the thylacum, that means the female sexual organ is having a specific character in upper portion, which is having a specific spine. So that is the species specific character for this group, Metopinopsis commensalist groups. And for male animal, the distal regions slightly extended lobe will be there. That is the major character for Metopinopsis commensalis. So this is not a commercially important species. At the same time, rarely it is available in the ornamental trade. So another one pinnate species, Parapinus investigators, which is also commercially important into the deep sea fishery. So the body will be very shiny and pinkish in color. The species specific character is the rostrum. That means the rostrum will be very short, which is having 8 to 9 small teeth on the dorsal side. So that is very specific character. A very short and a little curved in downwards. And at the same time, that specific character present in the sexual organ too. So the petasma will be subrectangular in shape, but it will be symmetrical. That means both the side will be evenly uh, not extended. It will be equal in size. So based on these character, we can identify it is the Parapenis investigator is one more species, almost the similar color to the Parapenis investigators, but the major variation is there in sexual organ, especially Patasma and Telecom. So here you can see that major difference. This is a slender and subrectangular Patasma, but here that is a little broad and little broad Patasma, very short also. And Telecum is having semicircular and shape with trilobe structure. See, here you can see that this is the semicircular in shape of the Telecum with trilobed structure. So this is a specific character for this species. When you see in the investigators, there is no this kind of structures. Only depressed halo kind of structure only is present. So that is the difference between these two species. Then another one species from family Solnasridae. So these species also very important to the commercial fishery. The species having the short rostrum, at the same time many more character in the carapace itself, especially that hepatic region is having a strong depressed sulcus, which is starting from the dorsal to branchial region. So this region we used to call as branchial region. The very strong sulcus, that means depression is present, which is starting from dorsal to in down position. So that is a major character. Also, one more specific character is there. So before the rostral teeth, there is a strong depress on the dorsal regions. That is a species-specific character for Solnosera melantho. So the based on the depression, we can identify it. This we don't want to see that the Patasma and Telecum and all. Only the one's character is enough to identify it as Solnosera melanthe. That is the strong depression on dorsal region of carapace. Coming to the next important species is Solnosera xt. Comparatively, that Solnosera species very easy to identify compared to other Aristidae and Pinidae group, because the very species-specific character present on the carapace itself. So easily we can identify it is in species level too. So here to the Solnosera xt, one of the deep sea shrimps and commercially important species too. Which is having a specific character on the carapace. The specific character is inverted L shape of the brachiocortex sulcus. That means that is a sulcus is present in this is the brachiocortex regions, which is having inverted L shape. So the strong sulcus is present in brachiocortex regions. So if the species having this character, so we can identify easily that is the Solnosera xt. So next species from the Solnosera is Solnosera anacton. So rarely occurred in the fishery, which is having specific character also in the carapace, especially rostral regions having a very minute hair-like structure, especially in the adrostral region. There is no hair-like structure on the carapace, but specifically the hair-like structure will be present in adrostral region only. So this is very specific character. At the same time, that brachiocortex sulcus is present little strong. So these two characters are very important for that anacton. So if you see these two characters, so we can identify it, this species from anacton. One more specific character is coloration. When you see in the fresh condition, the abdominal segments having a 3 different reddish dot. It will be there in the second and fourth and sixth abdomen. There is a red dot will be there. So based on the coloration also, we can identify that Solnosera species. So another one species, Solnosera pectinata, it's a close related to the Solnosera annecten, but however, the major variation is there in the rostrum itself. The rostrum will be a little long for the pectinate annecten, but pectinata is very short. At the same time, the major difference is brachiocordic sulcus. The sulcus is present very prominently, but here slightly it is present and very short. So this is the major difference between these two species. So if you see these two character, we can segregate to our species where it is matching with these two species. So another one, Solnosera species, the different genus Hymenoponius equalis. This is also important shrimp in the fishery. The major specific character is rostrum angle, especially that the rostrum is straight and upwarding. At the same time, the angle is 45-degree angle. So when the rostrum is like this angle, so easily we can identify it is Hymenoponius equalis. And another genus specific character is two epigastric. That means this region we used to call as epigastric, because here only that gastric region is present. So because of that regions, we used to call as epigastric tooth. So other Pinnae group and other Solnosera groups having only one epigastric tooth, but Hymenoponius groups having a two tooth. So if you see these two teeth and some gap with the rostral teeth, we can segregate that is Hymenoponius species. Yeah, coming to the next Caridian group. So one of the commercially important group, especially in the Caridia, the two genuses are very important to the commercial fishery. The one is Heterocarpus. So then specifically Heterocarpus chani is one of the important species in the Indian coast. However, many of the species from the genus in worldwide very important the commercial fishery, frequently harvesting from various region of the world. Specifically in that chani is the major specific character is present in the carapace itself. So that it is the rostral teeth, it will start from the middle of the carapace itself. But if you see some other group, there is no dorsal teeth on the carapace, but it is starting from dorsal region with the strong teeth. So the 4 to 5 teeth will be there in the dorsal regions. At the same time, another important character is this carina. That means a brachiocordic carina and antennal carina, which is extended near to the end of carapace. So it will be very prominent and very thick. So based on these two characters, we can segregate it is the Heterocarpus channel. So this species is very important in the fishery too. So here in Indian local market used to called as takali, because in takali means it's a tomato. Because in fresh condition, these shrimps look like a tomato. So in local fishermen used to call it as takali. So another one very closely related species is Heterocarpus udmosoni, which is easily identified specific character present on the third abdominal segment. So this also rarely coming with the Heterocarpus chani, but the species identification we can do very easily, because that rostral teeth starting near to the orbital region. But when you see in this Heterocarpus chani, the rostral teeth starting middle of the carapace. So that is one major character. Another important variation is third abdominal segment having a hook-like structure on the dorsal regions. So So based on this structure itself, we can identify it is Heterococcus woodmusoni. So this is the major difference between these two species. Another one important genus is Plissonica. Especially in our Asian countries, these species are commonly distributed. Around 97 species are available, and most of the species are commercially important and frequently harvesting from the marine environment, especially in deep water region. So all the species are mostly distributed in deep water region only. So coming to the species character for the Plissonica crossicandis, again that rostrum is having that specific character, that is the dorsal regions having a saw-like structure, which is having some 40 to 50 small teeth will be there. Then ventral region having some 30 to 40 small teeth. So that is the species specific character. So if you observe these minute teeth on the rostrum, you can easily identify it is the quasigrandis. So these also commonly distributed in Arabian Sea and Bay of Bengal. So next one is Plissonica semilevis, which is also close related to this Plissonica quasigrandis, but it is differing that rostral teeth itself, because in dorsal regions having the teeth pattern only 7 to 8, but other rostral length, other abdominal carapace features are very similar to quasigrandis, but the major variation present on the dorsal teeth itself, that is only 7 to 8. When you see that quasigrandis, that is 30 to 40 minute spine will be there. So that is the major difference between these two species, which is also distributed along with the quasigrandis. So but it's a rarely occurred with the quasigrandis. Though, yeah, another one important crustaceans that is from coral environment. So Anclearis brevicarpus, one of the important species in the marine ornamental trade, because which is having a colorful body and peculiar behavior pattern. So the especially in the body is having peacock tail structures in the tail portions. So in the ornamental trade, we used to commonly called as peacock tail anemones. Also, this species is associated with the sea anemone, especially for Heteractus magnifica, which is commonly distributed in Indo-Pacific intertidal regions. So especially in the marine ornamental trade, these species are selling around $10 to $15 per animal. So per single animal is selling in a very good price. So at the same time, which is having a colorful morphology. So therefore, that hobbyists frequently buying these species, and which is having a specific behavior pattern too. So here, especially this coral associated shame, we can identify based on the color pattern itself. We don't want to see other morphological features. Based on the color pattern itself, we can identify the species is which species it will be, whether Anclearis brevicarpus or some other group, because each and every species having a specific color pattern. This species having the specific color pattern is the brownish and white dot on the tail and abdominal regions. Yeah, that another one important marine ornamental species is Europaradilla andamburoni. This is commonly distributed in the Indo-Pacific regions. When you see in the fresh condition, it will be very transparent body. And at the same time, it's having that colorful texture, which having a red and white dot over the body. So it will be attractable color. At the same time, this is cleaner shim. That means the shims having a symbiotic relationship with some other grouper species. That means in the coral environment, the Europadella is doing a specific ecological role. That role is cleaning activity. That means the species used to remove the parasite, waste tissues, and some other mucus from the host animal, especially in the husk heel and some other grouper species. The host animal used to allow these shrimps to remove those kind of waste from the mouth and dorsal region of the host animal. So this is the very important species, also is a highly prized animal in the marine ornamental trade. So which is commonly distributed in the Indo-Pacific waters. At the same time, this group having is identified as a cryptic group because recently the scientists are observed that a similar kind of morphology and at the same time the coloration of the species, many of the region is very similar, but genetically it's different. So, they called us, it's a cryptic group and having many number of species from different region of the world. So at the same time, recently we also described one of the species from Europaridella, which is almost close coloration with the Undip. At the same time, morphology too. The slight variation is present on the this abdominal region, but genetically it's differed with this Undip, which is having high genetic variations. So later I will show you that how much variation is present between this Undip and one more our species from the Indian water. So that in coming slide I will explain it. Another one important interesting species from Lismata amboensis. This is commercially very important species and very colorful, and this also selling in very high price, and most of the hobbyists frequently used to buy this species because these species it will be very attractive in color. In the aquarium tank itself, it will show a peculiar behavioral patterns, so therefore which is showing more attraction to the hobbyists. At the same time, based on this color pattern, especially that reddish and white band on the dorsal region of the body, we can identify it Lismata amboensis, because each and every species having a specific color pattern on the Lismata group. Especially, we don't want to see that other morphological features. Only if you observe that color pattern, we can identify it. So another one important species is Lismata hochi. This is also ornamental species, so rarely occurred in the trade. So recently we observed from Indian water too, but it was commonly occurred in Western Atlantic regions. So the color pattern of the species is abdominal regions having the transverse reddish band. The bands are contains many small red dot which is making a transverse band. So if you see this this kind of band pattern on the over the body, so we can segregate easily that it is a Lismata hochi species. So this also is a transparent body with a reddish transverse band. So based on this character itself, we can identify this species belongs to Lismata hochi. Yeah, so this is the classical taxonomy of some of the important, commercially important species. So hope it will be helpful to see that different varieties and variation between these species. So Dr. Professor Norma, shall we start that molecular identification next session?
I think, what about if we discuss the first part? Make a discussion with the audience for the first part of the conventional taxonomy of the of these all very attractive crustacean. What about that?
Which ones? That grouper variations?
Yeah, yeah, yeah. Just give them some break by discussion.
Okay. Yeah. Sure.
Yeah.
Yeah.
Okay. Well, now I address the audience regarding what presented by Dr. Poos. He presents so very many and various kind of crustacea, either from the intertidal region or the deep sea crustacean. I would like you to raise your hands. I mean, those audience who maybe have something or to discuss with him, with Dr. Poos, please. Okay. Here we have 145 participants, which is very good. Anybody would like to ask something about the first part of the talk? Please raise your hands. Yeah, I mean, well, whilst waiting for people, I mean, the audience to ask you question, could I please start it? I have a question to use, maybe one or two.
Yes, please.
Yeah. I can't remember the name of the genus, but the name of the species is Woodmasoni. The one it has
Woodmasoni,
a
yes.
hook, yeah, a hook in the dorsal of the
Yes.
carapace. Not the carapace, I mean in the body.
Abdomen.
What is the, yeah. This is very interesting. What do you think this is for? Is that for a reproduction or is that for hanging around to something like a playstone?
Yeah, still they didn't. Yeah, still they didn't find out that what is the role in the biology still now, because which is distributed in deeper in water. No, so the most of the biology is still not studied in detail about the deep sea groups. Many of the morphology is having a different structures, but we could not conclude it because that distribution pattern is very deeper and benthic regions. So it's need to study. So what purpose it is exactly that morphology helping to the species still not at, studied in detail.
Yeah, but this is a benthic species. I mean, I can't see they have the walking leg. Is that only the pleopod? This with Woodmasoni? Oh yeah. Sorry.
The
That's in there.
pleopod is there. The same time
in the stick, yeah.
walking leg also is there, but the shrimps can walk and swim very fast. So the especially compared to the lobster from Reptantia group, that means the lobster can walk, but they can't swim. It will be distributed only in the benthic region, but coming to the shrimp, it can walk and it can swim in very fast. It can move from the pelagic region to benthic to pelagic regions. So the distributional horizontal and vertical distribution will be there for a shrimp group especially.
A further question, please. I mean, according to your observation, Pushji, are these animals, I mean, the deep sea crustacea, could they swim for a long distance? Or they just swim just to escape something and then they go down back to the benthic?
Yeah, sometimes they can go and hide it, but in biological way, there is that each and every group having some migration pattern. So it has to migrate in very long distances, especially these deep water species is to move very long distances, because recently we studied the stock structure of the Aristeus alcoki. The study showed that the species are having stock unit is a single stock unit in both Arabian Sea and the Bay of Bengal. So that study is suggesting that the species are having the migration pattern over both the sea. So that even genetic studies show that mixture of the stock. So from that we came to know that the species can migrate from one region to other regions periodically, but we don't know what time it is migrating and what time is coming back. We don't know that.
Yeah. Okay. Thank you very much. I mean, there is somebody raised a question. I don't know the name, but it's a she, I think, from Sinta. Dear Dr. Paramasiva, very interesting talk. Could you please explain to us what the structure that make difference between deep sea shrimp and shallow water shrimp? Thank you.
Yeah, that is actually interesting questions. That the major difference is there is no morphological variation between the deep sea and shallow water. So we can't conclude it a specific morphological character for only for deep sea and shallow water. But the other way we can assume that based on the morphological colorations. When you see in deep sea animal, mostly very reddish in color. But in shallow water, those kind of species, there is not a reddish in color, but slightly muddish in color. So the color difference will be there between the deep sea and shallow water. So based on the coloration, we can assume that the species belongs to deep sea or inshore. But coming to that coral associated, the coloration totally is different, because the each and every species are associated different invertebrate. If suppose one species associated with the sea anemone, the shrimp used to get the color from the sea anemone color itself, because especially the Tanclearis brevicarpus associated with Heteractis magnifica. So in that sea anemone having the similar kind of color. So this coloration which is helping for to hide the species from the predator. So especially most of the species having different color, but in coral associated we can't segregate based on the color pattern, based on the depth too.
Okay. Okay, is that answer your question, Miss or Ibu Sinta? Please raise your hand if you have a question for the first part of the talk. Yeah, thank you. She said thank you. Yeah, actually, I would like to. Yeah, I would like to further the question, why all those deep sea animals are normally they are red in color, whilst actually the spectrum of the sun ray, the red spectrum, the infrared is only very shallow.
Yeah, because all the species are distributed in benthic regions, so the light penetration will be very less. So therefore, these deeper water are mostly red or blackish in color, because of that light penetration is very less. No, so that is the major reason for this coloration of the.
So it's like they lack of the infrared, but they develop the red instead.
Yeah.
Okay. I mean, no more question for the first part.
Maybe that classical taxonomy always it will be bored to the student. I know that.
But I think that's quite very much interesting because it's not very difficult. You can see most of them just by normal eyes. Anyway, I think 11:13 in my time now. Should we start for the second part of the talk?
Yeah,
Pushi,
yes,
please.
started.
Yeah.
Yeah, next part is the molecular
Yeah.
identification of crustaceans.
Yeah.
Actually, the for fish genetic studies is to study in different hierarchical level for the conservation purpose to make a effective management statement for particular natural resources. We have to study in a different level. So that is based on the species and population and individual level. At the same time, the genetic studies are directly used for direct consumption. That means the food security in the fishery trade. At the same time, the molecular identification providing information for the description of the new species in a particular regions. Also, it's helping for the conservation of natural resources, especially identifying the endangered species and very important stock structure of particular species, also which is helping for accurate identification. But because many of the species having the species complexity, that means the similar morphological character and similar colorations, when it came to know that we can't identify through only from classical way. So where that molecular identification is helping to identify a particular species, whether the closely related species are similar or different species. So this, especially in this kind of identification, the molecular identification highly used. So here, so we can see that what is the advantage of a molecular identification. So, and the traditional way where it is lacking for the identification of particular species. In molecular identification through different gene barcoding, we can identify it in exact species level, also in cryptic species, and even a deformed tissue sample, and processed meat and canned meat, and even again larvae sample also we can identify through the molecular identification. But in traditional way we can't identify all this level. So only we can identify through one through that individual animal, through the morphology. So others are we can't identify, especially in the cryptic species. So both species looking like similar morphology, similar color patterns. So those time, the traditional morphology itself, we can't identify that species. So how many species are present in this group? So there, the molecular identification helping a lot to identify with how many species are present and how much variation is present one group to other. So this is the major advantages of the molecular identifications. So next, yeah, generally the identification of species through the DNA sequences we used to call it as barcoding. That means that identification of particular species through a short sequencing of particular gene, we used to call it as DNA barcoding. Especially the barcoding gene cytochrome oxidase 1 is very important and frequently used in worldwide for the identification of animal, various kind of animal species, because worldwide scientists are observed that particularly the cytochrome oxidase, which is a mitochondrial gene, so very important, at the same time showing that species specific variation, species to species. At the same time, it has to amplify in the PCR very good way. So therefore, all the scientists are suggested and made a that suggested that the gene cytochrome oxidase is a very so frequently used, and at the same time having the genetic variation. So therefore, that is the barcoding gene nowadays frequently used. Um, here we given the one example, that is how the species are varying one species to other species. Here that DNA base pair we used to call it as barcode because the variation present in the base pair itself. So you can see here I given the one example, species 1 and species A and B. So where the base pair variation present in between these two sequences, so those kind of variation only making the species variations. So this is the overall DNA barcoding workflow. So we used to collect the sample, at the same time the metadata is to collect it, and the DNA extraction will be carried out, and PCR amplification is to carried out with the specific primer, because each group having a specific primer, especially for shrimp groups. So there is this particular universal primer is developed by Falmer et al. So which will be commonly used for crustacean groups. So after the amplification, the DNA sequences will be done. Then the data will be stored in the public portal. It will be helpful for the future identification, although it can we can access in worldwide too. So here all the kind of metadata will be there, biological data, even the specific color images, and the DNA sequence and protein sequence of specific species we can upload with the DNA sequences. So for nowadays there is a specific site is there, that is a barcoding of life system that we used to call as bold system. So that specifically for a species identification, now all the barcoding data is stored in this database, which is linked with the NCBI database too. So here we can see that overall DNA barcoding pipeline, which is having different steps, almost similar to the earlier step, but here little more steps are there, that here, especially in the bold system, which is having a specific colorational image and detail of the voucher specimen detail, and so which can be useful to the different regions of scientists for the publication. So after the sequencing, so the sequence will be uploaded in the bold, at the same time NCPI2. So the sequence will be there in both the site, so we can access it, and we yeah, after the submission we used to get the accession number, which will be used for that development of articles or publication, whatever it is. Yeah, that I told you, bold system, DNA barcoding bold system. This is the homepage of the database. Here all the variety of database will be there. So here whatever you sequences, simply you can blast it, you can identify your species, so where it is matching, how much variation and how much similarities present, that detail also the database system is to give. At the same time, it will provide a coloration of the images too. So even voucher detail also used to get, because all the detail are stored in this database. So when you are blasting, easily you can identify the species. Here they are keeping the database in different level based on the animal and plant and fungal. So based on our species, we can blast and identify that sequence, that the sequence is what will be the species through the blasting. Yeah, this interesting thing is the worldwide there is different variety of scientists nowadays involved for doing the barcoding of life in different animals. So these are all the very important barcoding projects are going on for mammal group and marine barcoding group. So many projects are going on. So these are all the very important groups nowadays is going on. The database is there in bold system too. Yeah, for here I included one example of molecular identification of Aristeus species through different gene sequencing. So we have done 4 different gene sequencing. So where it is showing very good molecular variation to the other closely related species. Here we have calculated genetic divergence, and at the same time we just observe that how the relationship are there between the closely related species. The genetic divergence show that, especially in the CO1, very high genetic divergence between the species. That means between the species, we used to tell that interspecies variations. That means between the species. So it showed that more than 14% of variations. When it is the intra-species variation, that means between the individual of particular species, showed around 0 to 3% of variations. So here, especially this Aristeus group showed the more variation between the species, because very high variation was observed, especially Aristeus to Virula showed 43% of genetic variations. So that is very huge, because and especially the Aristeus group showed the more variation between the species. So the next one example is we have studied integrative taxonomy of some of important deep water peanut shrimps, and we have observed that phylogenetic relationship too. So here around 15 species were analyzed. So which are all the species from deeper water regions, one species from Aristidae, and some other species from Pineidae group, and the many species from Solnasridae groups. So that phylogenetic relationship is showing almost similar to the morphological relationship of these groups. So that the clade is indicating that Aristidae is so far to Pineidae and Solnasridae family. At the same time, especially the Pineidae showed between the species variation starting from 16 to 20% variation, but in Solnasridae showed 90 to 24% between the species showed the variations. Yeah, based on the integrative taxonomy, we have identified a new species recently, which is collected from the intertidal regions that we named as Actinimenes koya. The Actinimenes is a genus name. The koya is we honor to the local community people. There is one important specific local community people are there to we thought to honor those kind of communities. So therefore, we given the name to the species name Actinimenes koyas. So the while identification, we found that morphological variation, at the same time the genetic variation clearly denote that. So from the Lakshadweep Actinimenes species is a new to the science and having a very good genetic variation. So here you can see that the clear-cut clade pattern is observed. At the same time, very good genetic diversity. 18% are present with the closely related species. Yeah, so next we will see some brief information about the ecological role of the shrimps. See, we came to know that most of the shrimps are commonly used for food consumption and or marine ornamental trade, but the species indirectly is doing some very important duty on the marine environment or aquatic environment, that is which is actively involved in the food web. So where it is the consumer, at the same time is a major decomposer in the benthic regions. So that is the one of the major role for the shrimp group At the same time, already I told you that many of the shrimps are having a symbiotic relationship with many of the vertebrate and invertebrate animals. So doing that specific cleaning activity to many of the fish species and coral and other crustacean too. So I I met her told that this is the coral associated shrimp Lesmeta humbensis. This shrimp is to remove the waste and infected tissues from the host animal. At the same time, the host animal is to allow the shrimps to enter inside of the mouth. So during this cleaning purpose, so calmly it has to stand. The shrimp is to go inside and it has to remove. And the similar kind of activities were observed in Europrodilla species too. Even the most of the sea anemone associated shrimps too is to remove the waste from the tentacle of the sea anemone. So this is the major ecological role of the shrimps in the environment. Yeah, further we will see about that potential of the shrimp resources. So especially first we will see about in fishery sector because the shrimp is mostly nowadays very important to the commercial because fishery, because the world consumption of aquatic food is increased to 20 kg per capita, and also the international trade is denoting that in capture fishery producing 90 million ton, and which is the economic performance is 141 million USD dollar, and in aquatic production is showed 122 million ton and around 300 million USD dollars performing in the international trade. So here is in the trade, especially that aquaculture, the 60% of shrimps are produced through the aquaculture system. There, the few of the countries are highly involving for the production of the shrimps. Example, Thailand and Vietnam and China. And the remaining 33% of shrimps were captured through the wild environment. In particularly in India, around 1.5 lakh hectare are used for the shrimp farming, where mostly they're farming with the two important species, Penis pinemi, that is white leg shrimps. Another one is Penis monodon, that is game tiger shrimps. Also, the nowadays there is a one central government institute, Central Institute of Brackish Water Aquaculture. They actively involved for standardizing the breeding protocol for that one more pinnate species, that is Pinus indicus. So that they standardized the breeding and farming protocol and started promoting to the local farmers. So that is the third candidate to the aquaculture system. Coming to the capture fishery, if you see that Pinnae group, the 5 variety of families are involving in the capture fishery, but in Caribbean families, only 3 families are involving in the capture fishery. So these are all the species occurred in the capture fishery. Only that Aristidae and Pinidae family is the major resource for the capture fishery from the deep sea resources. So remaining species are rarely occurred in the capture fishery. So these are all the Caribbean groups, so especially Plesonica and the Heterocarpus genus are very important to the commercial fishery. Rest of that Glyptocarangon and Pacifidia are rarely occurred in the catch. So next important application is the marine ornamental shrimps. Already I told you that the coral associated shrimps having the colorful body structures and having a peculiar behavior pattern, example dancing, movement and other cleaning activity, so which make more attractive the hobbyist. So therefore the many of the hobbyists used to buy that this animal with a high cost. So that cost range is starting from 9 to $100 per animal. Especially that Lismata species are frequently collected from the coral ornament, and they're commonly available in the ornamental trade. And the rarely occurred species, that preclinious species, that is emperor shrimp, so a highly priced animal. That a single animal used to sell around $100. Especially in the marine ornamental trade, recently we have reviewed that around 150 species are available in the trade, and especially the genus Lismata and Stenopus are the most demanded species, which is commonly available in the ornamental trade, because which is to collect more frequently. Even millions of specimens are collected in annually in many part of the regions of worldwide. So these are all the different variety of ornamental shrimps, which is commonly distributed in intertidal region of most of the Indo-Pacific regions. So especially these Senatorangos and Rincho senators are very attractive species. So these species used to called as camel shrimp because of the coloration of the animal. So these are also a highly prized animal in the ornamental trade. Yeah, this is the overall ornamental trade cycle. So how the animal is collected, how the transferring to hobbyist, and how many internal internal marketing is there. These are all overall structure of the ornamental trade. So in sometime is to sell directly from the fisherman, and sometime is intermediate will be there, and sometime it will be breed through captive propagation and sell through the community aquaculture developments too. So this is the overall ornamental trade cycles of shrimps. Yeah, finally, so we see that important of the resources, and finally how we can conserve that shrimp resources, and how we can manage the sustainable management, and through the the control harvest. Generally, actually, the fishery sector, the fishery sector, and many things are used for food production. It's creating employment, recreational opportunities, and cultural identity. Even in recently, the FAOs listed that around the fish production was absorbed 114 million tons during the 2020. They noticed that around 58 million people are involved in the fishing operation. The economic performance was absorbed 115 billion US dollar during the 2020. So because it was observed that a rapid increases of human population over the year, so the demand automatically increased in considerably. So that's why the world consumption of aquatic food increased to 20 per capita. So see, you can see in the graph during the 1961 that world consumption of aquatic food range is very less. So you can see that how the gradually increased into the 20 kg. Coming to the shrimp groups, that recently in Florida government has noticed that during the 2016, 2.3 million number of Lismata individual are harvested from the coral associated regions, and they found that the stock depletion of Lismata was observed. So now they're taking care and making that management regime is based on that species conservation and management of the natural resources. For that conservation, there is a biological and ecological parameter of the particular species is always needed to make effective management regimes. So therefore, the different biological parameters always required. So example, recently we studied that stock structure of the capture fishery. One of the important fisheries, Aristeas alcoki, through the morphological way using trust network morphometry. So we studied the trust morphometric analysis for Aristeas species along the Indian coast. The study showed that the overall the Indian coast, both Arabian and Bay of Bengal, showing a single stock along the Indian coast. The similar study, once again, we confirmed with the molecular study using microsatellite primers, which is also denoting that the species are commonly distributed in Arabian Sea and Bay of Bengal with the single unit. So with high genetic diversity, the genetic diversity showing the indicating that the species having a having a very good genetic diversity and indirectly indicating that stock is having a sustainable way. So so that thing we concluded that the stock still it is in good conditions. So the based on the biological parameter, we observe that the species is in good condition in the wild environment. Yeah, the any other ways there to conserve the aquatic species? Yeah, there is a way. It is that the increases of aquaculture in for cultivation of marine shrimps, also the coral associated shrimps too. So especially in India, in National Bureau of Fish Genetic Resources has been taken initiative for development of breeding protocol and creating awareness on marine biodiversity and conservation of especially for ornamental shrimps. They standardized breeding protocol and developing the community aquaculture system in specifically in Lakshadweep regions. Here that shrimp farming, there is two institute are actively involving for development of protocol for peanut groups, especially Central Institute of Brackish Water and Central Marine Fisheries Research Institute from the Indian Institute are actively involving for the standardizing breeding protocol of many of the marine species and promoting that aquacultural developments to the farmer level. So not only aquaculture, some other way also we need to follow that is the continuum monitoring is required from the wild environment, and we have to give hands-on training to the coastal and highland communities, which will be helpful for the sustainable harvest and conservation of the particular species from the particular regions. And at the same time, there is a data tracking is required for the wild captures, and also we need to avoid that journal catches too. And finally, we have to create awareness to the fishermen in common people, even though the retailer too, we have to create the awareness, and at the same time we have to look on the non-indigenous species and their invasive nature and the coastal environment too. We have to observe that. Finally, that these are all my contribution to the related topic. So altogether, recently we have added some of new species and new distributional record to the marine fauna of Indian region, and we have standardized the breeding protocol for some of the important marine shrimps too. That is Thor hynensis and Anclearis brevicotilus. Even we have created awareness program and conducted in different regions of the Indian coast, especially Lakshadweep and Gulf of Mannar. Even in our previous institute, there is a National Bureau of Fish Genetic Resource. So started a community aquaculture unit in the Lakshadweep for promoting the ornamental shrimp cultivation and promotion into the community for a livelihood development and conservation of the natural resources. So this is my experience on this shrimp diversity, please.
Right. Thank you very much for the very interesting two parts talk, Gusti. Yeah. I think now,
Thank you,
yeah, I think some queries already in the queue for you. For the first, I think I see somebody, Pak Hengki. Pak Hengki, dipersilahkan. Okay, where's that, Pak Hengki?
Yes.
Okay, Pak Hengki, yeah. Please, you are the first. You raised your hand. You're supposed to have a question. No, I think you're still muted. Yeah.
Thank you for the chance.
Yes.
Dr. Paramasivan, you mentioned that China is a big three producer of shrimp.
Yes.
On your slide, 1 of 3. Well, I know that the water quality in China is not so good in average. My question is there any negative impact of China shrimp that people eat? That is my first question. And question 2, do you mind to share the minimum standard pH or COD water quality for the shrimp? Thank you.
Yeah. Actually, yes. There is a problem with the preservation here. India also faces the same issues in international trade. Because the most of the captured fishery from the deep sea, because they used to go in a multi-day trawler. So while coming to the landing center itself is to spoil it. So that is the major drawback in the capture fishery. So the same problem is facing India too. That
Sorry, I'm talking in bahasa.
Yes.
Saya minta tolong dimatikan semua yang tidak sedang dalam diskusi dengan narasumber. Terima kasih. Okay, please.
Yeah, because of that, the trade level has decreased even China and here too. So because that spoilage will create that insecure to the food content. Yeah, it's creating a negative impact to the trade too.
I see. Then last week I just shared. Last week I got the information that from the environmental health Undip that the negative impact of water can cause Alzheimer's. So it's very horrible. Do you think that can happen soon? Because the water pollution in China become worse. And actually, I'm going to join collaboration research with the China University. One of the China University is about water and air pollution. What do you think about that, Doctor? Thank you.
Exactly that, that water quality of the Chinese waters, I don't have that much information because I have to see that thing. Yeah, the water quality, it will affect the fishery definitely. Sure that the quality of the product definitely it will be affected. Yeah, that I have to refer it as. I will refer and get back to you that.
Yes, okay. Thank you. That's it. Thank you, Ms. Norma.
Yeah, thank you, Pak Henki. But by the way,
Thank you.
I
Please.
forgot to ask those audience to introduce themselves. Would you please introduce yourself, Pak Henki?
Okay. Actually, my name is Henki, and I am associate professor in previously in Malaysia, University of Malaysia. Now in University of Bina Dharma, and also I am guest lecturer in Undip. Also, I'm a friend of Dr. Frida.
Okay.
She's,
I'm sorry.
It's all right.
Yeah. I'm sorry that I don't know you. I didn't
it's all right.
recognize you.
And my research is more on sustainable development. Sustainable tourism development is one my project that invited by top talent in China. Hopefully this December I'm joined there for 5 years. It's a specialist in water and air pollution. This is
water and air pollution.
Yes.
Good.
This is a very what do you call it? Silent killer, I think. Silent killer for us. It can be give a negative impact because we eat and drink, and all of them is from the water, and the balance is from the air. So that's why from the beginning, I'm not so familiar with stream, but I'm familiar with fishery and also management and policy. So I'm not asking you about policy. Actually, I want to know, is there any environment policy in India? Actually, I have several friends, for example, from India, also analyze about water pollution there. I'm wonder, is there any policy in water treatment in industry? Maybe this is an option question, but it's not a must. Maybe it's actually this is good if you mentioning this research is under the policy act number blah 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 year blah blah blah. So I can easily to ask you in what point that you do this analyzing, because it's still in general. If I watch from the start till the end, actually I want to. I hope and I wish you can mention in term of certain policy that you done your research. That's it for Dr. Purus and also Prof. Norma. Thank you.
Yeah. Thank you, Professor
Henki.
Later on. Yeah. So another part from Dr. Pus that probably about what would you like to hear, the topics of something more related to your topics. But anyway, thank you very much. Yeah.
You're welcome.
Oh, well, we move to the chat here. I think I have a chat. I think I have a chat from a college.
Professor Norma, can you give me 2 minutes?
Yes. Yeah.
Yeah.
Would you like to? Okay.
Yeah.
Okay.
Yeah, 2 minutes.
Yes, please.
Excuse me, Prof. Norma. We have a question from Asigit. Can I share or not? We have one question from Masigit.
Yeah, we are waiting for Dr. Bush to come back to us.
Thank you, Prof.
Sorry for the inconvenience.
You're back now.
Yeah, yes.
Sorry, this is without break, Dr. Pus.
Yeah,
Anyway, we have, you okay now? You okay?
Yeah, I'm okay.
Okay, good, great. Yeah. Somebody in the chat asking about this. Here's one of the colleagues from my faculty. Great presentation, Dr. Paramasivam. What obstacles are often encountered when sequencing genes, and what is the most economical type of ornamental shrimp? Thank you.
Yes. Yes, actually, recently, we identified the Eurocardella species, which is one of the important marine ornamental shrimps. That is a very close related to one more species. So the almost all the morphology is similar, but in morphological way we could not find out the variations between the morphological features. But when we are using two different genes, especially for 16S and the COI gene, so when you do the sequencing, we came to know that there is a two group in the shrimps. So that is having a very great genetic diversity. So altogether it showed that there is a one new species is there with that group. So we found that the species is new to the science, and same time the minute variation on the abdominal segment. So there's such a way the genetic study is helping to find out that species identification, especially for a cryptic species identification. So the molecular tools greatly is helping for the identification of the these kind of species. That even many other Lismata species are also having the species complexity. So especially from Dr. Baisa from in Florida, the recently has identified the some two species from one group of animal. So in commercial ornamental trade, they used to identify as a Lismata species, one common species. But yeah, when Dr. Baisa used to know that there is a variation, minute variation is there between the species, and which was confirmed through the molecular way. They found out around three species, I think he found out from the group itself. So from that way, that molecular identification is helping a lot.
Okay. Yeah. Yeah. Okay. Thank you. Anybody, please raise your hand, or you may write in the chat if you have some more questions. Akura. Okay. Well, I mean, it's a little bit strange because people prefer to raise questions through the chat. Anyway, I will
Yeah,
do
actually, one that is one interesting the way it is actually. Our group also, we are working on it. Any other way, molecular or morphological way or ecological characters are available. The available is there to segregate this marine and some other inshore water shrimps. So we are trying to find out, but still there is no specific variation between these based on that environment. So even recently we made a meeting with our head. So that my head also is interested to find out the variation between these environment. So hope in future we will find out some interesting things for especially for the variation on this group from different environment.
Okay. What about the trust network morphometry?
Yeah, the trust morphometry will show that variation between the population. So, but it won't show the variation between the ecological conditions, especially different species. That morphology will differ, totally will differ. We can't conclude that this morphology species to species. Especially in the trust morphology will give us some idea to the differentiation between the population, particular species. So even sometime between the species, it has to give a very good variation, but between the environment, especially the ecological condition, it won't show much variation.
Any more question, please? I was wondering if you have ever
one more thing
analyzed
actually.
the chitin.
One more thing I want to share that actually the one more thing is the wild animal and the farmed animal, the growth variation will be there, but there is no that morphological shape variation, because the morphological variation will take much more time to show that the standard variation to the farmed things and the wild animal. So that is the stark variation. Maybe it will take much more time, around some 100 to 200 generation will take to get that kind of variation. So in the short period, it won't show that variation. That the growth variation will be there, but the morphological shape variation, it won't be there.
Okay. Good. Yeah, just a very small question from me, Dr. Pus. I was wondering if the shell component in between the deep sea and the intertidal species specimen, is there any different in that? I mean, the chitin content of the shell of the shrimp. Have you ever read, or perhaps you did it yourself in your laboratory?
Actually, one of our one more group is working on that, especially that component on the shell from the deep sea and inshore shells. So comparatively, deep sea animals having the more novel component in the waste stages. So especially in the shrimp wastage from the deep sea, It's having a very high quantity of antioxidant component. Then some other anti-inflammatory component. Recently, Dr. Kajal Chakravarti from Central Marine Fishery Institute from India, he has published many of the components from these shrimp wastages. Not only shrimp wastages, so other deep sea animal too. It's having a different variety of novel variety of components are there. Yeah, it's it's having a more component to the inshore. It's having a very good novel components are there. Even while handling that Aristeus shrimp in my lab, there it's having some of extra oil content. So maybe those content may be having a specific some other therapeutic application. It will be there. We have to find it out. So, from my knowledge, that especially this Aristides species is to live in very deeper and deeper. So maybe have much more component. So which will be it will be useful for that future therapeutic development too.
Okay. Okay, so it's very interesting, getting more and more interesting. Yeah, we have somebody here. I don't know, it's a miss or a mister. Sarah Fanan. Could you please open the camera?
Is there my head of the department from CMLRI?
Great. Could we see him, or is that a she or a him?
He. Yeah.
It's a he. Here you are. Please, you raise your hands. Dr.
Thank you. Thank you. Very good morning to all colleagues.
Yes.
It was an interesting talk by Dr. Purushothaman, and also I actually hear you all calling Purush, which is very interesting to me because we do Purush as Purushothaman. First of all,
Purush, yeah,
it is
Purush.
it is very interesting. Actually, his talk actually covered various discipline. I was actually knowing him only as an taxonomist of crustaceans, especially for the shrimp, but he now proved beyond actually a doubt that he is also actually capable of dealing management related issues. He has covered taxonomy, he has actually covered molecular taxonomy, he has also come up with some management strategies, he has also answered certain questions related to policy. So I think actually I'm seeing him as a first time a multifaceted person. So first of all, actually, thank you, actually, the organizer that actually letting me know the various multifaceted nature of Dr. Purush, as you call.
Dr. Purush.
Okay.
Thank you so much, sir.
Okay. It is an interesting, to see people are actually asking several questions. I do have some questions about actually, particularly the assessment of the population of Aristeus group. I understand he has actually gotten wide data collection, especially covering the entire Indian EZ. But how about actually the same distribution with regard to other parts of the world within Northern Indian Ocean? So, for example, Aristeus, you said actually it is the same population, found on both Bay of Bengal as well as Arabian Sea. How about this species distribution in other parts of Northern Indian Ocean? Are those actually population found elsewhere also is the same group? And it's also interesting because we tend to believe fishes are actually more migratory in nature, but not the crustaceans per se. They don't actually move forward. But how do they actually maintain the same species? How do they actually disperse? And with regard to Indian waters, we know but how about actually the this distribution of the species in other parts of the northern Indian Ocean? And I think it is a question to be answered by all the actually countries within this region, because the management strategy, whatever that we adapt for the India, should also be actually applicable to other area because if it is found to be a straddling stock, we should actually have an uniform management strategy for the entire region. So I would be interested to actually understand whether this population of this particular species is actually also spread across the other areas as well. Thank you so much.
Thank you so much,
It's
Dr. Saravanan G. Dr. Bruce.
It's a
Dr.
very
Bruce,
interesting one,
please.
sir. Actually, we have studied limited samples from Indian coast, especially in Arabian Sea and the Bay of Bengal, but the species is commonly distributed in Gulf of Aden to Andaman Sea too. So the further study is required to know we have to study in detail about Gulf of Aden and even the Pakistan coast here this side Andaman. Nicobar coast also we have to study. Then only we came to know that the overall distribution of the shrimps, whether all the sampling location, whether it is denoting a single stock or a multi-stock, we have to study in detail. This study is conducted only from limited sampling, but further studies required. Then only we came to know whether the species are having a single unit or any other multiple unit are present in the Indian oceans. Because why my answer is why the species are having the single unit from the Bay of Bengal and Arabian Sea, because both the water is mixing together at the same time. One of the scientists, he has worked in Aristeas group with labeling of animal. So he found that the species, especially Aristeas species, having a habitat used to migrate in long distances. So in the Mediterranean Sea, I think he found that Aristeas antonitus having the habitat used to migrate in long distances. So therefore, he also suggested that maybe that is the general features for the Aristidae family, because the other deep sea animal also used to migrate in long distances. When the water is mixing, the species used to migrate in long distances. So the similar way we also conclude, because while the monsoon period, there is a mixing of water is happening in Bay of Bengal and Arabian Sea. So maybe which make more convenient for the long distance migration for the species. So maybe that is the region. These sampling denote that the species is single stock, even though it's required to study in other regions, so especially Gulf of Aden and even that Andaman Nicobar region too. So where also that the species are reported in higher level.
Yeah, thank you, Dr. Purushothaman. I think it's a very
sir.
interesting topic. I also actually urge the organizer, if any such possibility for cooperation on this research area of this type of nature, that would be helpful in general for the entire region as well as for the whole humanity as such. So, please actually work out the possibility for bilateral collaboration or multilateral collaboration on the taxonomic research, particularly focusing on the crustaceans, if not for all crustacean, at least for the shrimps. Thank you so much.
Yes.
Thank you so much, sir.
Thank you very much. Yeah. Well, actually, one of the purpose of conducting this small international seminar like this is because we are developing our network locally, nationally, internationally with every single agency or student or individual who has who would like to join us or who has an interest in such a topics. Thank you ever so much, and we are waiting for you. Or maybe I can get you contacted through Dr. Puss.
Yeah.
Dr. Puss.
Thank you very much,
Yeah.
Professor Norwa. It's also I'm grateful to the Dr. Dean of the Institute. And thank you very much, one and all. So we will see in
Yeah. Yeah,
as soon as
yeah.
possible.
Okay. Yeah, yeah. Well, there is one more question, I
Yeah.
think, from my college, Dr. Bambang Sulardiono, to
Yeah,
Dr.
yeah.
Paramasivam, provide information about the genomic DNA isolation stage and following what protocols. I mean, this
Yeah,
is
there is
using.
two different common protocols. So it will be helpful. One is salting out protocol. Other one is phenol chloroform method. So these two methods, which will be very cheap and best, you will get a very good quality of DNA. Suppose if you are getting any trouble, you can go for that kit protocol, especially blood and tissue kit from Quagen company. It will be very good for that isolation of the DNA for some critical sample, long preserved sample or deep water sample. Some deep water sample we can't get the very good quality from manual protocol. So that in blood and tissue kit, it will be helpful to isolate such kind of DNA isolations.
Okay. Yeah. And to your experience, what is the best primer for, or any difference between primers for deep sea and the shallow water specimen, please?
The primer is for CO and G. Palmer is a very good. That is a universal one.
Yeah,
but suppose
Palmer.
Suppose it is not amplified, you can go for one more primer is Crusta. Crusta for only crustacean, so it will be helpful for specific amplification. It won't show that variation between these two primer. So when you're analyzing the DNA sample, it won't be a problem. But some specific primer, if you're going to use species specific, means we can't do that phylogenetic analysis with available gen bank sequences. So where you will face that sequence based per variations. So, but these two primer, especially universal primer, Palmer et al. At the same time, one more primer Crusta. So it won't make this kind of issues when you're analyzing phylogenetic analysis. Then one more gene, that is 16S gene, so which will be very good for that phylogenetic analysis. Especially, it will show very good relationship between the species compared to CO1. The CO1 is very good for the species identification, which will show that high genetic variation. But in phylogenetic view, that 16 is very better for that.
16. Okay.
Yes.
Yeah. Okay, great. Yeah. Lots of things we have been discussing and studying from you and from all the audience. I think, what time is it now? Okay. We've been talking for quite some times, and thank you ever so much for your presence, for your expertise, and for your kindness to explain things to us. And most of the audience, I think, are students. So I have a very great hope that this talk today will widen their knowledge and their passion to such a study like you. Thank you ever so much, Dr. Purusottaman Paramasivam.
Yeah, thank you. Thank you very much, Papaji.
Yeah. Well, I asked Master of Ceremony, I ask Pak Kuku to continue the
Okay, Prof.
agenda
Norma.
for today.
Okay, thank you so much, Prof. Norma, as our moderator for today's webinar. And thank you so much also for our Zoom speaker, Dr. Push, who delivered the insightful presentation about the Unveiling Stream Diversity. Thank you so much, Dr. Push. Hopefully we can meet in person, maybe in India, or maybe you
Yeah,
come
sure,
to
sure.
Indonesia, especially come to Semarang to our
Yeah, sure.
faculty. Okay, move on to the next agenda. I would kindly asking Dr. Aninditya, as head of postgraduate program Aquatic Resource Management, to give a certificate of appreciation to Dr. Push. Maybe the committee will help us to show the script.
Okay, thanks Mr. Kuku, and also thank you Professor Norma for your time and your kindness to be moderator for this session. Yeah, thank you Prof. Norma, and also Dr. Pus. Today we are very glad to have you in this seminar. Thank you very much for your time and sharing your knowledge and experience. Hopefully, we still can keep in touch, yeah, in the future and maybe make other collaboration, yeah. So here we have a certificate for you. Please
Thank you
stay
so much.
healthy and success.
Yes.
Thank you, Mr. Koko. Thank you, Prof. Norma. Thank you,
Yeah.
Dr.
Thank you.
Yeah, you're very welcome.
Okay, thank you very much, Dr. Anin, as a representative from our department who give the certificate to our speaker today. And here we are at this moment, we have reached the closing part of today's webinar. I really do appreciate for your attendance and excitement in this today webinar, despite the busy schedule that you guys have, especially in this weekend. Not forget to mention Prof. Triwinarni, Ibu Dr. Sianti, Ibu Kurun Ain, Ibu Dr. Aninditya, and all of the lecturers, committee, and staff that cannot be mentioned one by one to spare your precious time today. And maybe the committee will help us to share screen again. We have some attendance form. You can kindly check the chat box column in the Zoom or in the live chat on the YouTube, because the committee already prepared for the link below, and for the certificate and the lecture presentation, you can access by this link. Yeah, you can also download your certificate and the lecture presentation by access this link at aquatic.fpik.undip.ac.id or at mmsdp.fpik.undip.ac.id. Okay, next slide, please. And don't forget to check out our online platform. We have an Instagram for additional information, and don't forget to subscribe to our official YouTube to get notification about our next webinar. And also you can visit our website at aquatic.fpik.undip.ac.id to know more about our department. And you can contact us to our email at seminar.msp.undip@gmail.com. Okay, see you again in the next webinar. I, as Master Ceremony, apologize if there are any mispronounce and or mistake in delivering today's session. Keep update and check our social media for upcoming event. I'm Kuku Prakoso as today Master Ceremony signing out. Thank you and see you in another session. Goodbye. Assalamualaikum warahmatullahi wabarakatuh. Stay safe and healthy.
Thank you, Mr. Push. Thank you ever so much.
Yeah, thank you, man.
Very good topic.
We'll meet
I will
shortly.
ask again. I will
Yeah.
ask again. Anytime you have.
Thank you, Dr. Pus, Prof. Norma. Thank
Yeah.
you so
Thank you.
much.
Yeah,
Thank
you're
you.
very welcome.
Thank you.
Mhm.
Good afternoon.
Okay. Uh-huh.
Kak Ida, apa kabar?
Indonesia is an archipelagic country with one of the world's richest reefs and longest coastline. This gives Indonesia an immense potential in the fisheries and marine sector. The Faculty of Fisheries and Marine Science of Diponegoro University has been among the leading marine research institutions in Indonesia, since it was first established as Fisheries Department in 1968 and officially became a faculty in 1994. The Faculty of Fisheries and Marine Science is located in the Central Java Province. The main campus, where main activities take place, is located in the capital city of Semarang. The second campus, also known as the Marine Science Technopark and Coastal Development Laboratory, is located in Jepara Regency. The vision of the Faculty of Fisheries and Marine Science is to become an excellent faculty in the field of fishery science and tropical marine in the year 2020. In pursuing its vision, the Faculty of Fisheries and Marine Science missions are to provide a high-quality education in the field of fisheries and marine science, engage in scientific research and publications, produce intellectual property rights, provide community services in fisheries and marine science field, implement a professional and accountable system, and empower students' activities to achieve the complete graduate qualifications. There are 6 departments in the Faculty of Fisheries and Marine Science.